We think a lot of the diagnostic tools that are out there in terms of blood tests to find patients actually omit Alzheimer's disease from FTD have come light years in the last 5 years. People are now shipping blood tests to show your risk for potential likelihood of Alzheimer's disease or dementia. If you can find patients earlier in the life cycle and have a really good and thoughtful way of intervening with your drug, there's a better likelihood to show a benefit in those patients.
Chris, I'm so excited to be having this conversation. Thank you for joining me.
Thanks for having me, Brandon. Look forward to it.
Yeah, of course. For the audience, Chris is the CEO and co-founder of Psilera. Psilera is doing some really interesting work applying psychedelic treatments to neurodegeneration and neurology more broadly. We've had a lot of conversations on this show about the applications in psychiatry. And when I learned about the work that Chris was doing, I just had to have this discussion. But Chris, maybe just give us a thumbnail sketch of your career and how you started working on this topic.
Sure. So by training, I'm classically a chemist, particularly a natural products chemist. So really a lot of my PhD research was focused on finding new drugs from nature. Did a lot of fungal research as a PhD student, looking at, anti-infectives, new oncology medicines, things like that. It's actually a really good starting point for drug discovery. About 70% of all FDA approved drugs literally start out as a natural product from nature. Even GLP-1s, let's say the drugs that everyone's talking about, it started out as a compound found from Gila monsters, so reptiles, and they were able to fast through use of this GLP-1. So, when you look across the psychedelic medicines now, as we really know them, things like psilocybin, DMT, even LSD, really natural products or naturally derived molecules themselves. So, there are things that I studied and referenced in my dissertation as a PhD student. So always kind of near and dear to me in that respect, because I think there's just a ton of therapeutic promise here. So after graduating, actually, I started with a company called AltMed. It was a startup founded by a former pharmaceutical executive from MedImmune. And really their goal was to become a premier medical cannabis company. And my role, I was their first scientist, ultimately becoming their chief scientific officer, was to devise new drug delivery technologies around cannabinoids. So really what my role was is developing an IP portfolio of transdermal drug delivery systems, meter dose inhalers, nebulizers, very pharmaceutically relevant delivery options where, obviously smoking is not something the FDA would look at very, very fondly. And really kind of my role expanded from there more into operations, developing products and out licensing opportunities to expand the brand. And kind of oversaw a lot of manufacturing, personnel, and as a natural products chemist, these things are very kind of things that I've studied my whole career doing. So kind of found myself at the right place at the right time. And it was kind of during that same period where, there was some early research happening with things like psilocybin and MDMA that kind of piqued my interest and actually allowed me and my former colleague, so Dr. Jackie Von Salm, also a co-founder of Psilera, we did our PhD research together, worked at AltMed for a bit. So it was just kind of, again, being at the right place at the right time, reading the tea leaves and understanding that this really does have the potential to help a lot of people.
Yeah. So having spent a lot of your career thinking about natural products, what was it that you saw in the data or the trends that were starting to come out with psychedelics that kind of gave you the conviction to do this? And then in particular, I think the kind of target indications you've chosen are quite different from maybe what we commonly see. So tell us about the thinking there and the story.
Yeah, I would say one of the big milestones for us to really jump in and start Psilera was the approval of Spravato or esketamine from J&J. So literally within a few months after that, we founded the company, and we think this kind of opened up a playbook from the FDA of there are now regulatory steps to take that you can approve a medicine even as untraditional as these are. But I mean, going back to, I mean, the 1950s, 1960s, some of the data that was being generated from LSD across a number of areas for mood disorders or addiction. I mean, there really was a lot of promise. And then, ultimately there was a lot of regulations that kind of banned this research. And, some early researchers in the early 2000s kind of repicked this up, and you're seeing the same trends. And it's not like an N=1 kind of study. It's very routine and very, we think the data was very regimented. And for us, we looked at these as, again, as natural products chemists, what types of ways can we create better drug out of this knowledge. And for us, that was really around drug formulations and also creating new drugs that don't have the potential side effects that psychedelics do. And, you talk about, neurology and neurodegeneration. I mean, again, we think this is a really big underserved patient population who really struggles with a lot of mood disorders. About 90% of all dementia patients have depression and anxiety, all these things. And we think the first generation psychedelics are probably contraindicated here. Whether it be increased risk of psychosis in these patients. Obviously, there's memory and communication problems that can impede sort of the normal set and setting profile. So we think by removing these psychedelic or hallucinogenic effects, you can actually make a more tolerable treatment specifically designed for these patients.
Yeah, absolutely. I want to get into that in just a moment, but actually before I do, because you mentioned the esketamine pathway, I'm too curious to not to ask. Dan Carlin, who we've had on the show from MindMed, talks a little bit about how the legalization of cannabis might have hampered the ability to kind of pursue the more rigorously tested therapeutic effects. I'm curious, having spent time in both of these spaces, how does that resonate for you?
I actually fully agree. For us early on, we met with some people who really kind of catalyzed the formation of Psilera, became investors in the company. And, when you look at cannabis, basically the public opinion really surpassed the scientific evidence. And we think that's kind of backwards, especially for, we think drugs with immense promise. Also, I think the way that psychedelics work in the body and the effects that they produce really should not be as publicly accessible as things like cannabis. So, for us, it was, okay, we need to set the bar high from a regulatory, a clinical, scientific data standpoint. And then, the public opinion can kind of come thereafter once the physicians and the regulators validate it. And we think this is probably the best way to create, sustainable, educated, patient population out there. There's obviously probably pros and cons to either side of that. But yeah, I mean, I think there's immense promise here, but we need to let the data kind of lead the efforts, and then let access and patients kind of come next.
Are you worried at all about some of these psychedelic compounds following a similar pathway?
I mean, obviously there are some jurisdictions now that are allowing it, and I think there's definitely ways to do it. There's good guardrails that you can put around it for safety. Primarily is where I'm focused. I think the efficacy is what it is, but yeah, I'm cautiously optimistic about some of these things. I mean, I'm all for patient access, and that's why I think a lot of our model around Psilera is how do we increase access around these types of molecules. I mean, we think removing psychedelic effects, ultimately, these could be first line treatments or best in class treatments. Patients with dementia probably don't want to take a traditional psychedelic. We think there's probably a lot of people out there that don't want to take a traditional psychedelic. So how do you increase access? And I think, some of these early movements that you're seeing is all about doing that in a way that, maybe does it in a more cost effective or at least in a more timely manner. So yeah, I mean, I'm not really advocating for either side of it, but obviously watching and, just hoping that, good outcomes and, you're not getting the bad headlines that kind of stymied progress here for a long time.
Yeah. Yeah, absolutely. Okay. I want to change gears and talk a little bit more about Psilera. Your lead asset is, PSIL, SIL. How do you pronounce that?
Yeah, SIL 006.
SIL-006, um, uh, and you're studying a group of uh, conditions around neurodegeneration, specifically with non-hallucinogenic psychedelics. So maybe just like catch me up on the thesis here. Like how is this supposed to work? What's the hypothesis?
So early on in Psilera's formation, we spent a lot of time going through literature. So Jackie and I are both chemists. So looking at structures, we developed a platform. It's a computer-aided drug design platform where we can make molecules very quickly, put them in proteins of interest. So for us and psychedelics, it's really serotonin 2A. So I'll say 5-HT2A. And this is really the psychedelic receptor responsible for the effects. And, there are a number of case studies for us to look at and kind of develop an early set of molecules that we tested first in computers. Then we hired a team of chemists to then make these molecules and do kind of the more classical preclinical models, looking at animals and receptor testing to kind of validate what our initial hypotheses were. And ultimately, this research landed us on SIL-006, which it is a psilocybin derivative. It's a new compound. Very small modifications were made to psilocybin in order to produce this molecule. But what we've done is effectively reduce the hallucinogenic potential. So we're not seeing any of those side effects in any of our animal models. We've shown very rapid mood benefits in things like depression and anxiety in preclinical models. And then, as you mentioned, we're looking in neurodegenerative areas. So we've done a lot of models in humanized tau. So tau is obviously a protein responsible for dementias, Parkinson's disease as well. And with animals with these tauopathies, you're going to see, traditionally cognitive impairments and sleep disorders. And that's obviously what you see in humans as well. So we've shown the ability to reverse a lot of these cognitive decline in animals. We've also shown better sleep patterns. So you come up with these things called sundowning syndrome, where your sleep schedules are effective. You don't get as good of sleep patterns, but we've shown really actually a rescue in these animals with SIL-006. So our lead indication for this molecule is called frontotemporal dementia. Affects about 60,000 patients within the United States and almost about 1 million worldwide. And it is a rare dementia, but when you look at the underlying pathology of the disease, we have a really strong thesis that, number one, we can provide a rapid benefit to patients in terms of symptomatic improvements, like I said, with mood and sleep and cognition. But ultimately, we think the way the drug works and the specific pathologies actually around serotonin 2A, that we can actually reverse or slow down some of these traditional declines that you see with patients with FTD. So this is kind of our first test case, but ultimately, if we see benefits there, we're looking to expand this into other dementias and related Alzheimer's indications as well.
Gotcha. So this is not not just a symptomatic approach. This is also potentially like a disease modifying therapy.
Yeah, for us, we're a small biotech company, so to go purely after disease modification is obviously very tough, and it's a long road. And obviously, there's a lot of case studies out there of other companies that have tried this. But when you're looking at more of these symptomatic cases, which is really our step one, we can prove these benefits in 12 weeks, and this is in line with other psychiatric trials that are going on with psychedelics. So you can actually show these benefits much faster. Cognitive decline takes 12, 18 months. And, even then, you really have to power your study properly. You have to include a lot of patients just because of the heterogeneity of the disease. So we think by showing a symptomatic benefit, where there's actually a lot of traction in this area across big pharma companies. You've got a couple approvals now in agitation in dementia, psychosis in Parkinson's disease. There's a number of late stage trials that are really going after more of these symptoms as opposed to the overall disease modification. Ultimately, we think speed and efficiency is everything in this industry. So that's where we're starting. But ultimately, we are going to look at more of the potential cognitive impacts of the drug, which really do correlate to disease modification.
Yeah. So for longtime listeners of the show, I think folks will be familiar with some of the like underlying hypotheses or that kind of surround the kind of symptoms around mood disorder, sleep, etc. I think we've talked about that a lot. Maybe like let's go like a layer into the disease modification thesis. Like how are we thinking that kind of plays out?
When you're looking across, all of the dementia and Alzheimer's trials, I mean, really the primary endpoints to prove disease modification are showing a cognitive benefit or at least a reduction in cognitive decline. We know a lot about the pathology of say dementias, and again, that's kind of one of the reasons we like this area is there are biomarkers there that you can assess patients that have the disease. There's a genetic component too that's lacking from a lot of the traditional psychiatric areas like depression and anxiety. So we think these are going to aid us ultimately as researchers and translating better data from preclinical to humans. So, initially our focus really is number one on mood. Can we provide a rapid benefit there? And everything that we've generated to date and other clinical data really, I think is highly supportive of that. Secondarily, we will be looking at the cognitive impacts of the drug. More often than not, it's going to take longer to show a statistical improvement. So you're just going to have to monitor patients for longer. But that is something that we're building into our clinical development plan. Looking at things like tau specifically or other kind of protein hallmarks of the disease, I would say we're a little early in our journey to really kind of opine on that. But those are things that we are actively investigating. And, if and when we get new data to kind of support this, obviously that's an area that, I think holds a lot of promise. So if you can show a cognitive benefit or show some, molecular biology improvement in patients, ultimately that's the holy grail in this area.
Yeah, a lot of the kind of topic of conversation around psychedelics has been around this idea of neuroplasticity. At least within the mood disorders. I'm curious, like, help me map the idea of neuroplasticity to degeneration, or is that even not the right way to think about it?
I think in a broad paintbrush that it certainly works. When you look at neuroplasticity, basically it's really easier to study in animals than it is in humans. There are really robust ways to demonstrate this where you take neurons, you culture them in a petri dish and you look for new dendritic branches or new connections between axon. So that's kind of the definition of neuroplasticity. So it's basically just improving neural connections in the brain. And really this is the underlying mechanism across psychedelics, across neuroplastogens, things like ketamine, psilocybin, they all kind of have this similar mechanism of action all around neuroplasticity. And when you look at the hallmarks of dementias or neurodegeneration, it's literally atrophy or dying of neurons. So you can find a way to stimulate those neurons with a neuroplastogen, obviously you can start drawing conclusions there. I think it's a little too early to tell, but certainly that goes into the hypothesis of what we're trying to prove out. By stimulating these neurons, can you help reduce atrophy in the brain? And that's something that you can image in patients. So, there is a conclusive outcome that you can look at. We think there's a lot of different ways that you can kind of assess an efficacious pathway here. We're kind of looking at number one, can we improve the quality of life for patients and their caregivers? And I think this is really important that a lot of people overlook. A lot of people are looking for the moonshot. Okay, we're going to, solve Alzheimer's disease. Well, obviously that's a lot easier said than done. But if we can at least help patients initially, monitor them longer on the drug and seeing if it is slowing the disease progression, we think this is probably just a more sustainable, less risky way of going about the drug development pathway.
Yeah. Fascinating. Help me zoom out a little bit on this topic of psychedelics in neurodegenerative indications. You mentioned frontotemporal dementia. How do you think more broadly about the landscape of neurodegeneration and the applications of psychedelics therein?
I actually highlighted a study that was published recently. It was an academic study. I think a small number of patients, maybe 12 or so, with Parkinson's
I saw
disease.
that.
And they were given a 25 milligram dose of psilocybin, so a fully psychedelic dose of psilocybin. And they monitor these patients largely for safety. Like I said, there's an increased risk of psychosis there. There weren't really any anomalies. It is a small study, so there are some caveats. But really, I think the results kind of confirmed what at least we believe at Psilera is. Number one, there were very rapid benefits on mood across the board. And again, these patients do struggle with depression. Imagine getting this diagnosis and saying, well, there's not much we can do there. And this is a broad issue across many neurodegenerative categories. But also one of the hallmarks of Parkinson's disease is these movement disorders. And I think that was kind of where I was a little more surprised that there were benefits in the rigidity and increasing movements in Parkinson's. So, while psilocybin primarily affects the serotonin pathway, and most of the evidence in Parkinson's disease are with dopamine and dopaminergic agents, that was interesting. And there's definitely, again, I think a lot of promise here. It's very early days. Again, I think we're probably going to start to see more data in this patient population. I think there's a little bit of apprehension there. Certainly, as I mentioned, from a safety profile, there are, I believe, better ways to make therapeutic agents for these patients without the psychedelic effects. But this is certainly a good first step in the right direction. And at least to my knowledge, this was really the first, kind of clinical study and clinical evidence really supporting that hypothesis.
Yeah, I think that's the paper that I saw you post about that really got me interested in this topic. I'm curious, whose research in this domain are you most closely following, most closely interested in, and kind of staying on top of?
Yeah, for us at Psilera, I mean, really our primary focus now is in these neuroplastogens, these non-hallucinogenic derivatives like SIL006. So again, there's a lot of late stage research going on with first generation psychedelics. So there's really only a very small handful of companies that are doing kind of what we're doing. Certainly there's Delix Therapeutics. So they have their neuroplastogen now in a phase 1B in treatment resistant depression, I believe, or MDD maybe. So, one of their big findings was, okay, you dose their patients in phase 1 and they didn't see a lot of the psychedelic effects. So that at least goes to show that a lot of the preclinical models that everyone's running, it's called a head twitch model, where you dose some mice and you monitor the head twitches. So the more the mouse head twitch, the more potentially psychoactive the drug is. So at least that was sort of a de-risking milestone for, I would say, a lot of the other, researchers in this neuroplastogen area. At the end of the day, it all comes down to clinical evidence. Preclinical is great, but obviously, we've probably solved many more diseases in mice than we ever have in humans. So, you really need that clinical evidence. So, we're looking forward to bringing SIL006 to the clinic next year, and following the evidence of what's out there. But for us, I would say a lot of the focus in the serotonin 5-HT2A receptor, it's the most well-known drug target, I believe, ever. There's 20 plus drug approvals. And these molecules are largely antagonists, so basically reducing receptor function. Most of the psychedelics are agonists, so increasing receptor function. So it really goes to show that any way you engage this target is efficacious. So for us and the other neuroplastogen companies, it's all about finding a balance of, okay, can you engage the target without tipping the scales into hallucinogenic effects? And, obviously I think there's some skeptics to say, oh, you only need the psychedelic effects. Those are driving the patient outcomes, the efficacious outcomes. But when you look at the evidence, I certainly believe that there's enough of it out there to generate clinical evidence. We speak to a number of neurotherapeutic heads at major pharmaceutical companies, people much, much smarter than I am, all of which, they're really looking at this category very closely. They certainly believe that you can develop an efficacious drug without the hallucinogenic effects. but ultimately you need the clinical evidence to support it. So, we're still a little early in that journey, but, I definitely think the next few years are going to be really, really interesting in this space.
Yeah, absolutely. Do you anticipate there being a, let's call it like a second or third wave of excitement here around these non-hallucinogenic compounds? I know that this, let's call it first wave with the traditional or kind of first gen compounds is kind of, things are in phase 3 now. Are you anticipating like an upcoming wave as well as people pursue maybe the second generation?
Yeah, I mean, it's the clinical development, drug development pathways. History doesn't repeat, but it rhymes. So
Right.
let's take ketamine, for instance, and J&J gets to approval with treatment-resistant depression with intranasal ketamine. So now you've got a number of non-dissociative, non-psychoactive NMDA. So this is the way that esketamine works, is targeting this glutamate pathway. So now you've got a number of phase 2, phase 3 NMDA compounds that are non-dissociative. So basically you get one drug to the approval line. It's de-risking that drug target now for a number of researchers. And it took, maybe 4 or 5 years, I would say, after the approval of Spravato to get there. So, when you're looking at the next couple of years for drugs like psilocybin, like DMT, I fully believe there's going to be a wave of these non-hallucinogenic drugs in pivotal trials. It's just kind of the way things work. And, certainly this, I would say the particular area of these neuroplastogens, the non-hallucinogenics, those are primarily where pharma's focused. There's already been one big deal in this category, looking at these neuroplastogens with AbbVie. So, the commercial model, I think, of psychedelics is leaving some people scratching their heads. Obviously Spravato, that's a good case study, 2 hours, get patients in and out, no psychotherapy. It's $1 billion drug. So if you can fit your drug into that kind of framework, there's a de-risk model there. But for things with longer in clinic times or the need for psychotherapy, I mean, that kind of convolutes the commercial model. But neuroplastogens, you don't have the psychedelic effects, you don't have blinding issues in your clinical development. Ultimately, these drugs can be approved for at-home use, so that limits a lot of the clinic and the insurability of those first-gen molecules. So, it's kind of the way things work, and I certainly think that the next few years, again, there's going to be a number of these compounds in phase 2, phase 3 trials.
Yeah, absolutely. So take me a little bit into the patient experience. I'm so curious. You just mentioned, if I go in for let's call it like a psilocybin, a classic psilocybin like experience today. I'm in clinic for, let's call it 4 to 6 hours. And that ranges depending on the kind of compound here. For non-hallucinogenic compounds, what is it? I just take it and I go on with my life.
I mean, yeah, that's really the goal of what we're developing is, non-psychedelic, non-psychoactive compounds that don't really interfere with your daily life, only have the benefits that, we all think they have in terms of mood and cognition and sleep. So yeah, that's the goal is to develop just a typical medication,
like an aspirin.
A supply, take it home. Yeah. And, limited side effects, we think that is ultimately the holy grail. Again, not to use that term lightly, but that is the potential there, is to really harness the benefits of these psychedelics, but just removing side effects. And again, this is the clinical development pathway that pretty much every best-in-class drug follows. The first drug on the market is rarely ever the best drug on the market. So it's companies like us that really remove side effects to make it more tolerable or to make the commercial model better, increase patient access for us, looking into dementias. We think this is a great way to engage those patients. So, I don't think what we're doing is super novel. I mean, for good or for bad, but it's a pretty stepwise process in logical development.
Hey, having sat on the other side of the table on fundraising, I've gotten asked all the time, well, what about competition? Like, why wouldn't J&J just develop this? Or like, there are other kind of psychedelic like sponsors that are better capitalized right now. Why do you think that there is this, like, let's call it emergence of new biotechs developing these non-hallucinogenics, as opposed to the kind of folks that are developing the first generation, just iterating on their kind of assets?
I think big pharma is typically risk averse. That's quite frankly why the word neuroplastogen was used. So you don't have to use the term psychedelic. It's obviously, it can be a buzzword, but it also cuts the other way. So yeah, I mean, early stage research in this, the onus is really on biotech companies to show the evidence, and then the light bulb goes off. Pharma says, hey, look, there's enough data here now that we can really get involved. And I think AbbVie was a good case study a couple years ago. That was preclinical. There was no clinical evidence on these molecules. So they got in a little bit early, but from our conversations, we definitely think there's a lot of potential there. There's a lot of interest from mid to large pharma companies looking at this category. And again, it's on the small biotech companies to kind of prove out the model, show there's evidence. There's kind of the I would say the competitive landscape has thinned a little bit, which we think the best companies kind of emerge from these types of environments, and ultimately, they're the ones who can persevere and really kind of go to that next level. And, for us, we're looking at those partnerships. We think the buy-in from big pharma, they have the regulatory experience, they have the deep pockets to really usher these in to commercialization. And we think that's, beneficial for a company like us, especially when, the fundraising landscape is what it is. Those partnerships are obviously crucial for companies to thrive and survive.
It strikes me that the applications of these neuroplastogens in neurodegeneration have been maybe underlooked, or maybe maybe just kind of like overlooked. Relative to the psychiatric conditions. I'm curious, like, why do you think that is?
Again, I think when you're looking at it from a safety profile, there's obviously some red flags out there for degenerative patients with psychosis, memory problems, the set and setting profile that's, crucial to kind of inform consent and, preparing a patient for the psychedelic experience and the integration thereafter. If there's a memory problem, there's going to be issues, and, that can lead to agitation in these patients, or leaving them kind of worse off than where they started. So, I think there's been some apprehension for these first gen molecules to go into Parkinson's or dementia. I think that's why we kind of looked at it. It's okay. Can we take the beneficial aspects of these molecules, but remove some of the liabilities so we can find new patients who would benefit? And that's exactly what we're doing. I think there definitely is promise there. When you look at the mechanism, again, neuroplasticity, engaging those neurons, especially in areas of the brain. For us in FTD, it's all in the prefrontal cortex. That's exactly where our drug is active. So if you can increase activity in the regions of the brain that are otherwise atrophying in these patients, we think there's significant benefit there. And there is one molecule that's in late stage trials now, phase 3 for Alzheimer's disease. It's a non-psychedelic, but it engages a drug target called sigma-1. And when you look at most of the psychedelic molecules, they interact with sigma-1. And really what they've shown with this phase 3 data is reduction in brain atrophy, improved cognition, also the ability to kind of engage sigma-1 in sort of the brain's, mitochondrial to kind of actually flush out these bad proteins. So, when you look across, potential mechanisms and potential drug targets, I mean, we think there's a lot of, clinical evidence that we can point to of why we should target, these potential patients with our drugs. So, yeah, again, excited to show the clinical data, because ultimately that's where the rubber meets the road. But again, there's a lot of things pointing in that direction that we can really benefit some patients here.
What do you think most people get wrong when thinking about this space?
I think I touched on one earlier, that you need the psychedelic experience in order to have a therapeutic outcome. I think, sure, you can point to say, well, there's not enough clinical evidence to support it, therefore it doesn't matter. But also, there's a lot of smart people out there who are thinking exactly like we are at Psilera, we need to generate this evidence, because if you can develop a drug without these side effects, I mean, we're talking, I mean, it's a blockbuster many times over, just because there's immense unmet medical needs. I mean, people in rural areas who can't go to a physician and have a 4 to 6 hour or 2 hour experience. So, how do you reach more patients? How do you not, omit patients because there's a prior history of psychosis in their family, or, they have high blood pressure, so they can't take a psychedelic. So, we think really this is a logical step to take in the drug development process. And ultimately, if these drugs are successful, there could be best in class and first in class potential. So, again, there's risk there, but we think it's a risk very much worth taking. Again, I think your point to neurodegeneration, and we think this is an area that's highly underexplored. I mean, there's obviously been a lot of negative headlines in the past about, potential beta amyloid pathologies, and does it actually work? Does the preclinical evidence support, development of these? But when you're looking at neuroplastogens and drugs like SIL-006, again, targeting symptoms and mood really can benefit patients and their caregivers immensely. Anything above and beyond that is really just a cherry on top. And, we think there are a lot of evidence out there to support the translation of these drugs into humans. Another big area of pathology in these patients is actually neuroinflammation. And I think a lot of people don't understand how potent psychedelics are at reducing inflammation in the brain. I mean, like, incredibly potent. So when you look at biomarkers in dementia and FTD, things like IL-6, which is a cytokine pro-inflammatory marker in the brain that you can assess through blood. You don't have to do CSF or lumbar punctures. So this correlates to the disease progression. So the more inflammation there is, generally, the more cognitive decline you have, the more agitated and apathetic these patients are. So, these types of biomarkers are immensely beneficial in these neurodegenerative categories that, I think psychiatry ultimately will catch up, but, we think there's just much better guardrails around stratifying patients in neurodegeneration, and also having these biomarkers that certainly aid and de-risk the programs.
Sure. I mean, a lot of the conversations we've had on this show center around this idea that these subjective markers in psychiatry make the research particularly difficult. And as I've been having more conversations in neurology, it certainly seems like we're getting closer to being able to have a little bit more objective markers. What are the challenges, I guess, involved with running like these programs for these kinds of groups of patients?
In neurodegeneration specifically?
Yeah.
We're looking at FTD first. I mean, it is a rare disease.
That's
so,
rare.
In some ways you can look at that as, limitation. 60,000 patients in the US, though, is sizable. So we don't really expect to have, any issues finding patients and recruiting them. We think a lot of the diagnostic tools that are out there, in terms of blood tests to find patients, actually to omit Alzheimer's disease and traditional dementias from FTD, those have come light years in the last 5 years. I mean, people are now, shipping blood tests to show your risk for, potential likelihood of Alzheimer's disease or dementia. Things like neurofilament light chain, those are also being used as a blood marker to show patients with a specific disease like FTD. Again, we think these things are really going to help get a diagnosis faster, because I think that's one of the hard parts of neurodegeneration. I think there probably is a point where it's almost too late to find a effective intervention for the disease. I think a lot of research has shown recently that if you can get to patients faster, it's like cancer. I mean, if you catch it too late, it's too hard to slow or reverse the disease. And I think that's particularly, the case too with the brain. So if you can find patients earlier in the life cycle, and we have a really good and thoughtful way of doing that, and intervening with your drug, there's a better likelihood to show a benefit in those patients. So, I think those are, I would say, the current challenges. More may come up and down the road as we get into the clinic, but for all the reasons that psychiatry is difficult and, your endpoints are subjective, how do you feel? There's a huge placebo effect. Expectancy is an issue with psychedelics. The blinding aspect is also particularly challenging around psychedelics. So with our drugs, we think without having those effects, the blinding is still intact. So that gives regulators a lot more confidence in the data that we would generate.
Yeah, I was just thinking that this idea that like the functional blinding is so difficult for psychedelics, maybe is, I guess, probably almost definitely easier for kind of compounds that are non-hallucinogenic.
Mm-hmm. Yeah, I mean, on the functional unblinding, I think J&J and Spravato, that's the playbook that people need to follow. I mean, they got to the finish line of 84 milligrams of esketamine. I'm sure there's some unblinding in there. So, yeah, and you never almost want to be first in some of these areas because you can learn from others' mistakes. So, we know we're behind these phase 3 trials in traditional psychedelics, and we're going to learn along the way. And again, I think over time, the way that neuro has really, I would say, just kind of improved in terms of diagnostic tools and therapeutic interventions have gotten a lot better in the last couple decades. I mean, this area, especially neuropsychiatry, I mean, since the 90s, no one was really doing anything, but there's a lot of excitement, there's a lot of interest, and I certainly think this data around psychedelics is really going to kind of help improve the visibility here, and ultimately, it's going to provide more opportunities to develop better tools and better drugs to help patients.
Absolutely. Well, let's look to the future a little bit here. I saw an announcement for a major licensing deal with Atai. Congratulations. We've had Trini on the show. I know that they're a very thoughtful team and how they approach development. How are you hoping this to kind of come together and support the next phase of development for you?
Yeah, as I mentioned at the onset of Psilera, we kind of had two pillars. Number one was finding new molecules and creating new molecules like SIL-006, but also part of our programs were how do we effectively deliver drugs like DMT? So this molecule has no oral bioavailability. So most people were just injecting it or giving it IV. And, we think there are better ways to do that. So we devised a campaign, developed IP around, different technologies, intranasal delivery of the drug, transdermal delivery of the drug. As time progressed, we kind of had to make one bet on what clinical development program to follow. And ultimately that's SIL-006. But at the same time, we found a very like-minded partner in Atai. And, I think we both share the sentiment that DMT is an immensely valuable therapeutic asset. And they have their phase 2 program, VLS-01. It's an oral mucosal film with DMT. Again, non-invasive. We think there's a lot of benefits to patients there. So we found a synergistic way to work together where, they now have a better patent portfolio with our DMT patents. And, we have an economic value as well tied to their program. So for us, it allowed us to reinvest into SIL-006, generate more data on the efficacy side, on the safety side, so that we can get 006 to the clinic. And, for us, I mean, we think it's a win-win across both sides. So, again, like you, we think Atai is really well versed in this. They have a great team. I mean, Srini's a great guy. He's got a lot of experience. And I think what they're developing there is certainly one to watch. And, we're happy to work with them. And, we think there's a lot of potential for their programs.
Absolutely. Paint me a picture 10 years from now. How do you think this landscape looks? How do you think psychiatry is being practiced?
Yeah, I mean, I think it's going to be a lot better. I think number one, patients having optionality is everything. I think what Spravato ultimately showed is having a therapeutic intervention that works quickly is valuable. Patients don't have to wait 6 weeks to get better. They can get better in one day. I think what you're going to see fingers crossed from some of the data in phase 3 for psilocybin, DMT and some of these other assets is a similar rapid acting benefit to patients, which obviously, you want to get better right away. So that's going to be there. I think the durability of these first gen psychedelics are going to exceed that of Spravato. So I think having the optionality for patients and physicians to say, okay, well, these drugs didn't work, let's try something different. And I think that's going to be really valuable. I think looking, and that's only, I would say, a year or two away in the near term to get some of these new therapeutic options in the hands of patients commercially. For us in our category, again, we think that kind of creates a lot of downstream opportunities to create better drugs, these neuroplastogens. There's a handful of companies in our position too that are kind of preclinical, advancing into the clinic. So in the next few years, we'll be in phase 2 for FTD, and others are looking more at psychiatry. And, again, we think optionality is a good thing. I never really thought the industry could kind of go as well as it has in the last 5 years. I mean, we started the company in January 2020. The pandemic happened. Mental health became very much in focus. People were locked down. The news was always bad. So, of course, that created I think it opened a lot of eyes for the problems that are out there. I mean, 1 in 4, 1 in 5 people worldwide are having a mental health problem every year. I mean, that's a huge, huge problem. So the fact that, I think psychedelics are probably going to kind of open this landscape up a little bit more. I think it's going to open up more opportunities for other therapeutic options to get out there. For us looking in dementia, I mean, we think from a symptom standpoint, there's a lot that we can do, but, if there is the ability to slow the disease, I mean, quality adjusted life years are everything for patients. So, I think there are definitely a couple, neurological assets in late stage trials now that, the data is looking really, really promising. So, I think there's going to be a lot more interventions that are out there. I think the tools to assess how drugs are working, these biomarkers, platforms, stratifying patients, those are getting better. And the technology is getting cheaper, faster, smaller, more scalable. So ultimately, that's going to help assess patients, and finding them better and making sure they're responding to drugs. So, I mean, I'm, the golden age of psychiatry and neurology. I think we're not far off.
I think we've had a few conversations on the show about interventional psychiatry. I think largely due to some of the constraints around the delivery models. Does all that go away if these non-hallucinogenic compounds really, really work here? Do we not have to be thinking about interventional psychiatric clinics, all that kind of stuff?
Well, I think maybe one of the big questions around these neuroplastogens is what's the durability? We don't have really any eyes into what that is. I mean, we know, at least from a lot of the late phase studies now with, we'll say, psilocybin, I mean, 1 dose, 3 months out, 6 months out, I mean, there's still a lot of remission in those patients. For the non-hallucinogenics, we don't know if that's going to be the case, or if it's going to be more of a routine dosing. So, I mean, I definitely see a world where both of these classes coexist together. Again, it's about what patients and physicians want. It's like, okay, well, if you want to go through this, it's 1 dose and, 3-6 months out, you'll be better. And for some people, they might go there. For others, okay, let me try something a little more routine, but the side effects are more manageable, and fit more with my daily lifestyle. I don't have to go and find a clinic. So I definitely think, there's a world where these coexist. Or if someone takes a psychedelic dose and then maybe they have a neuroplastogen kind of maintenance dose to keep them in remission. There's a lot of different scenarios that, these all kind of coexist as one cohesive, toolbox for physicians.
Yeah, certainly. Sometimes we think about these kind of conditions as being more homogenous, whereas everybody's journey is a little bit different, and their lives and their schedules usually dictate what kind of treatment regimen makes the most sense for them.
Yeah, I agree 100%.
Chris, any Um, any last thoughts here that we haven't touched on? Is there anything that you wish more people asked you about the space?
Wow, that's a good question. I mean, let me think on this for a second. I think people are skeptical of this drug class. And, I think that's maybe not an unhealthy thing to do. But when you look at the evidence, I mean, I think it goes back to the point you said earlier, where kind of the hype cycle kind of maybe went far beyond what the scientific evidence would support. Well, I think the pendulum has kind of swung back the other way, in that people are highly skeptical of these drugs, even pharma companies, investors. And when you look at the scientific evidence that continues to come out, I mean, I certainly believe in the near term there are going to be commercial options out there that are available for patients. I think engaging this drug target, which is the most well-known drug target that we know of as researchers, it really doesn't take a lot of kind of thoughts to put together the potential of this category. And when you look at the unmet needs that are out there, yeah, I mean, everything comes and goes. Now everyone's talking about GLP-1, so mental health kind of gets pushed aside. And then, it's kind of this constant back and forth, but you kind of have to ground yourselves every once in a while and just say, okay, you got to keep doing what we're doing and staying resilient and continue to generate data that is going to change people's minds. And, generating clinical data and generating commercial NDAs and INDs of getting a new therapeutic option to patients is everything in this. So, I mean, that's been our singular mission from day one, is do good science, and everything else will kind of fall into place. So, good times and bad, that has not changed. And, we remain very well rooted to what our goal is, and that's to provide new options for patients. We say a new era in mindful medicine. So, we're very, very early in what I think will be a very interesting years and decades ahead in this space. So, hopefully there are going to be a lot more answers than questions in the not too distant future. But, I'm always here if there are questions to be asked.
Yeah, certainly. They say that progress happens slowly and then all at once. And it kind of feels like it's happening all at once right now, which is an exciting time to be.
For sure. I agree.
Well, Chris, I've so enjoyed this conversation. Thank you again for taking the time to have this discussion with me.
Thanks for having me, Brandon. It was a pleasure.
Absolutely.